Hello, welcome to the UCLA Center for Global Management Race for a Cure webinar series. I'm Jennifer McKinney, faculty at UCLA Anderson, UCLA Medical School. And with me, I'm happy to welcome an esteemed panel of industry leaders responsible for many of the early advances in COVID-19 treatment, as well as two leading key academic opinion leaders from both the UC system as well as Switzerland. First, I'd like to welcome Dr. Alan Wright, the Chief Medical Officer of Roche Diagnostics, James Larus, Professor and Dean of the Computer Science and Communication Sciences at École Polytechnique Fédérale de Lausanne. And along with him, we have Dr. Jim Rooney, VP of Medical Affairs at Gilead Sciences. And finally, Dr. Tom Coates of the University of California Global Health Institute and current anchor at UCLA faculty member. For those of you joining us this evening, I'd really like to highlight sort of three key themes in this discussion today. And those revolve around detection, diagnostics, and treatment. The recent FDA approval of an antibody assay and therapeutic treatment for COVID-19 has really launched some of the discussion today. We'll be hearing from both Roche and Gilead regarding those two products. And in parallel, technology, one of the things we're really interested in Anderson is the intersection between technology and industry, particularly healthcare. And so we'll hear how technology is helping to aid in the detection and tracing of COVID-19 and potentially the treatment. So with that, I'd like to maybe kick it off with each of our panelists, a moment to introduce yourselves to our listeners this evening. Special thanks to James, who's joining us from Switzerland, where I know it's 2 in.the morning. So thank you, James. I see some great pajamas picked out for the call this evening. So appreciate you joining us at this time of the evening. So why don't we kick it off with you, Dr. Coates, and we'll pass it on to the rest of the speakers afterwards.
Thank you. One of the things I wanted to talk about was the parallels between COVID and HIV. I was around, I was a faculty member at the University of California, San Francisco, 1981, 1982, when that was ground zero for HIV. And there are a lot of differences between COVID and HIV, obviously in the way the viruses are transmitted and in their lethality. But there are some interesting differences, and that is that HIV came into the United States at a time when the culture wars, like now, were strong, and when the administration did not want to deal with it. In fact, President Reagan didn't say the word AIDS or HIV until 1987, 6 years after the first cases were described here in San Francisco and Los Angeles and New York. With COVID, we have very much of a parallel situation with an administration not dealing well with it. I think it's important to take a moment and acknowledge what's going on in the streets of Los Angeles and many other cities, and that is the demonstrations going on to mark the murder of George Floyd. And it's connected to COVID because part of the discussion, part of the concern, part of the issues being discussed is that COVID is disproportionately affecting African-American communities. And this is just another part of the long history of our brutal history of dealing with African-Americans in this country. Happily, the happy part of the story is that HIV rewrote the history in terms of how to accelerate drug development, drug access, and access to vulnerable populations in two different eras. Last week, we marked the death of Larry Kramer. Larry Kramer was an AIDS activist, playwright, and he founded at least two activist organizations. He made extremely good friends with Tony Fauci, who you've seen a lot lately. And it was Tony and Larry and others like them working together to say, the normal speed is not okay. We need to accelerate development. We need to accelerate it, and we need to make it accessible to the people who need it the most. And that happened. It took a while, but it did happen. And that's the good news here, that in fact did happen. You see Tony Fauci in his role today, the same role he had in 1984, 1985. And the issues are the same. How can we accelerate the development and deployment of these medications? Unfortunately, I mean, I don't want to be a Debbie Downer, and I hope that the story is different with COVID. I remember in 1985, Margaret Heckler, Secretary of Health and Human Services, standing with other scientists saying we will have an HIV vaccine in 2 years, but we don't have one yet. And it took until 1996 to develop highly effective medications. So one of the issues I think that I'd like to bring up and make sure we discuss tonight is the fact that, well, acceleration is good. It may not be as rapid as we would like. I hope it's different with COVID. I really do hope it's different. We need to have this. The second big point that I want to mention, and then I'll stop talking, is the fact that in 2003, with strong bipartisan support under the administration of George W. Bush, the United States funded and started PEPFAR, the President's Emergency Plan for AIDS Relief. And that brought these life-saving medications to people in low and middle income countries, particularly sub-Saharan Africa, who would otherwise not have access to these medications. And the pharmaceutical companies were part and parcel partners of this effort to bring these medications to the people who needed them the most. And that, to me, is the issue of both bipartisan, but more important, global solidarity. And that's what it's taken to get us to this point in HIV, and we need to do the same for COVID.
Thank you, Tom. I think you make a great point about balancing speed, scale, safety, and societal benefit. Alan, why don't you also kick it off with an intro, because certainly Roche has demonstrated great speed in launching the Alexis antibody test.
Yes, thank you, and thank you for inviting me. I'm Alan Wright. I'm with the Roche Diagnostics Corporation, and we're part of the Roche Group, which also includes Roche Pharma, Genentech, Foundation Health, or Foundation Medicine, and Flatiron Health. Regarding the Roche Diagnostics, we manufacture a broad variety of instruments that range everywhere from the glucometers, those finger stick measures for glucose, to molecular diagnostics, which include sequencing solutions and PCR, which we'll talk about tonight, to the big blood analyzers that are in hospitals, to point of care solutions, and tissue staining. And with that, we provide, this is an old figure, but about over 17 billion results a year for patients. We don't actually do the testing, but we manufacture the instruments and reagents that go into that testing. We have achieved emergency use authorization for our PCR test for SARS-CoV-2 testing in March 13th. We have emergency use authorization for our serology, the antibody testing, on May 5th, which is for the measurement of exposure. And just 20 minutes ago, the FDA gave us emergency use authorization for interleukin-6, which can be useful in the management of what you've heard euphemistically called cytokine storm. We've shipped 9.3 million PCR tests to date, and more to follow. So back to you, Jennifer.
Thank you, Alan. Jim, can you follow up as well on your recent news and announcements?
Sure, yeah. Hey, Jim Rooney here from Gilead Sciences. I'm a VP of Medical Affairs. I've been at Gilead 24 years now, so quite a while through the development of most of our portfolio. Good fortune to work with Tom over the years, so we certainly recognize all the important work that has been done in that field, and we would like to echo as well what Tom mentioned, that the types of collaboration, innovation, and partnership that were put together between academicians, industry, government investigators, and funders addressing the HIV pandemic is exactly the same kind of collaboration that we need nowadays to address the COVID pandemic. So we certainly are very much in support of that. Gilead, for those of you that may not be familiar with the company, is a biotech company based in San Francisco. And our initial focus primarily on antiviral medications, and included a variety of those, including treatments for influenza, hepatitis B, hepatitis C, as well as HIV. And we had medications for both treatment and also prevention of HIV, including some of the first single tablet regimens one pill once a day for HIV, a triplet that's been then succeeded by several subsequent generations of pills. And then the first medication for prevention of sexual transmission of HIV, which is augmented by the development of Descovy. More recently, we've been involved in the addressing various different RNA viruses, including respiratory viruses, research programs on RSV, but also programs came out of our Hep C program, including for emerging looking back at SARS-1, at MERS, and more recently at COVID. And maybe later on I can describe how that evolved for us in terms of our ability to respond in a fairly rapid fashion to the COVID epidemic as it exploded here really across the globe. So I'll also say, in the concept of partnership and PEPFAR, that Gilead does have a global partnership with a variety of generic manufacturers that help us make our medicines available globally. And for example, for HIV, we had over 11 million patients that received one or more of our medications last year through that kind of partnership. So again, underscoring the importance of global cooperation and partnership to address an important public health emergency.
Thank you, Jim. And James, I'd like to kind of close out the introductions with you. Certainly for those that didn't have a chance to read the webinar invite, the technology that James has developed within his research is responsible for the platform that Google and Apple are now using for privacy-preserving proximity tracing. So James, can you tell us more about that specific platform as well as some of the other research that you're involved with in the space?
Sure. Let me start by thanking you very much for the invitation to be on this very distinguished panel. I'm the outlier here. I'm not a doctor. I don't do medical research. I'm a professor of computer science. I spent some time in industry and been a professor for a number of years. EPFL is the Swiss Federal Technical Institute. It's basically MIT, if you want to think of it that way, in Switzerland. I really knew nothing about this area 3 months ago, like most people, I think, in the world. But I did have a member of my faculty who is an epidemiologist, or as he calls himself, a digital epidemiologist. And when we all got locked up, got put in our apartments, he basically came to a number of us and said, we need to do something. And contact tracing is the most immediate need, and it can be brought into the 21st century. Everybody has a cell phone now. We can use cell phones to determine who you've been near, keep track of that, and when you're exposed to somebody, give you a warning that you've been exposed to somebody. The faculty I had in my department were fortunate. I was fortunate that a number of them were extremely concerned about personal privacy, which is a very big issue in Europe. And so we designed an extremely privacy-preserving contact tracing system, which Google and Apple subsequently picked up. And 3 months later, it is now shipping, well, it will be on about 1 billion cell phones throughout the world in a matter of a couple weeks from now. It's the basis for the Swiss app, which we also built and delivered last week, the first app that was delivered on the Google Apple platform. And there'll be a number of other ones following in Europe in the next couple of weeks, probably in the United States as well, and eventually pretty much in every country, because it's not practical to build a contact tracing app without the Google Apple platform. And I'm happy to say that we can do contact tracing with it, and we can preserve your personal privacy, which is extremely important for this particular type of app because it's a voluntary process to use the app. You have to decide that you want to run this app, and you want to take the basically the altruistic attitude that you will notify other people if you come down with COVID and you're diagnosed. You'll tell the other people that you came in contact with so that they can, again, do the right thing and go into isolation, into quarantine, and avoid spreading the disease further. And again, it's very much a participatory activity. It's something where people have to believe that the information that is collected on them is not going to be used for any other purpose besides contact tracing, and that their personal privacy is not going to be violated by using the app. And we're quite confident that what we've built actually meets all of those goals. So, thank you.
Thanks, James. I did get the primer in our pre-discussion on Bluetooth beacons. I'm hoping maybe you can share a little bit more of how exactly that all works with our listeners.
Sure, I'm happy to go into great detail.
But maybe just for myself, just as a disclaimer, also coming from an engineering background. But Tom, I think what would be helpful, particularly for listeners, is just we're certainly looking at treatment at the treatment side and the other sort of downstream pieces. What scientific studies are really needed to better understand just SARS-CoV-2 specifically, as well as treatments for COVID-19?
If I understand you mean just to understand how it's settling into the population and how we find. Well, there are two different kinds of studies. One are surveillance studies, where you attempt to determine just what is the prevalence in the population. Rudimentary studies here in Los Angeles County, the folks at USC have done that and came up with a prevalence estimate of somewhere between 2 and 7% of LA County individuals may have had or do have COVID. The problem is there's no one prevalence estimate. It really depends a lot on where you live and your exposure. So if you live in South LA or if you live in Panorama City, you're going to have a higher prevalence in those cities because that's where more lower socioeconomic people live. So that's one kind of study. You just want to kind of find out the spread, prevalence, where people are, and you can do that by mapping cases and mapping symptoms. And once the contact tracing app gets up, that will be very useful in sort of mapping where the clusters of cases are. The more important issue probably is to find out where the active cases are and then to work out from there. And this is, so it's not testing just the general population is not as effective as targeted testing. Following where the cases are, who the contacts are, testing them, and working out to find out where the clusters of cases are. The group at Harvard has done a lot of modeling on this, and what they say is you've done enough testing if your prevalence of infection is lower than 10%. And of course, that's going to depend on your population density, as well as the nature of the community and their propensity to be exposed to COVID. So that kind of gives you a sense. So I think in terms of prevention and control and treatment, the more important piece of that is to do this targeted testing and then targeted contact tracing out so that we can really identify and rapidly identify those who have been exposed to COVID and who may be symptomatic and transmitting it currently.
Yeah, thanks, Tom. That's a great lead-in actually to my question for Alan, because specifically, you guys have obviously done this amazing process as well as the new IL-6 announcement today, but this is 46 days, right? So you now have an antibody test. You also have some other products that you're in the process of getting ready to launch or have already launched. How do you sort of compare this to business as usual? And then based on what Tom said about sort of the fluctuation in prevalence and as well as the kind of change in clinical trial capacity in various geographies, how do you think about moving forward with these various assays and treatments that you have in the pipeline?
Okay. Well, you know what? First, let me give you a timeline. So it's December 31st of last year, and the World Health Organization declares an epidemic in Wuhan. By January 7th, we have a sequence from a lab in China. Roche is a global company, so we form our emergency response group January 8th, and we begin work. Now, know that this is done in a environment where there isn't free flow of biospecimens, so we cannot get biospecimens out of China. So our first work was done on using contrived specimens, spike specimens. The other thing we did to actually select the target, we used in silico modeling. The virus is about 30 kilobases long, and we use computer modeling to select the best targets to develop our probes to greatly limit the number of trials that we needed to attempt before we found the ideal set of probes. Also, something people really don't talk about a lot is the manufacturing, because know that, you know, we started out with hundreds of thousands, and now we're manufacturing millions of assays. So we're making this on an industrial scale, and there's a scale-up process that's involved, and these are done in bioengineered organisms, and they really don't care about our urgency. They grow at their own speed. And so we had to make pilot batches, and then quality check them, and then experimental developmental batches, and then finally production batches, and that took time as well. But, you know, fortune favors the prepared, and, you know, I couldn't be prouder to be part of this team, but they were able to do that in remarkable time. And the same thing with the antibody assay, similar aspects where, you know, very limited data sets. And one of the things that speeds things to market, and we're experiencing it now, is it usually takes us 3 to 5 years to develop an assay. We're actually doing this in reverse, where we have brought an assay or some assays to the medical community, and now we will continue to develop the evidence set to support the assay, the analytical evidence set and the clinical medical evidence set to support that assay. So all the clinical questions that people want to know, you know, does it predict immunity? And can we use it for identification for convalescent sera? We don't know because we brought it quickly to market. And then as far as the analytical set, there's a lot of further confirmation that we need to do. How does the assay work with people with, you know, high protein in their blood, or bilirubin in their blood, or, you know, abnormalities of their blood? We don't know that. We continue to work on that. So it's really sort of tearing the wrapper off of assay development, and the public gets to watch how an assay is typically developed prior to launch. You get to watch it on the market. But we continue to develop it and test it, and it continues to be a very high performing set of assays, and the parameters that we set for launch continue to hold up, and we're very pleased with that.
Thanks, Alan. And certainly, I would agree that, you know, we've heard about real world evidence, but this is definitely real world evidence in action, and will continue to evolve as we see how the FDA responds. You know, Jim, maybe to you as well. Gilead has shown amazing speed to market in launching what was previously an unsuccessful therapy in treating Ebola. Can you talk about sort of that process again, sort of you know, start to finish, and what the anticipated outlook is, either for treatment or, you know, other routes of administration, different patient segments? What is the sort of the plan there?
Yeah, I mean, as Alan said, I think our ability to respond antivirals for potential emerging virus infections, and it arose out of a similar program for other RNA viruses, respiratory syncytial viruses, and hepatitis C. The particular compound we're using now for treating COVID was not as effective against those particular viruses, but it did show activity. We went on to test it in collaboration with others, federal government, other collaborators who had available specimens and an interest in terms of exploring this, looking at potentially other important pathogens. Middle East respiratory syndrome is another coronavirus that arose in 2010, 11 or so, and was tested against that. And then obviously against Ebola, and had activity in the test tube and in animal models. In Africa, and compared to other treatments, monoclonal antibodies in particular, it was not as effective. It probably wasn't the optimal treatment for that disease, but there was still some evidence that it did have activity, potential activity, although obviously we no longer pursued it for that. However, as a result of that, we felt it was important to have a stockpile of medication available in the US in case Ebola happened to establish itself in the US. And so we had both a stockpile of about 5,000 vials, as well as, very importantly, a compassionate use program. So this is a program that allows you to make medications available on a case-by-case basis to patients who may have no other options, but essentially, are facing a life-threatening disease, et cetera, et cetera. So when the epidemic arose in China for SARS-2, it was clear to us that remdesivir could potentially have some activity here. We again began exploring its activity against that particular virus, which was shared many characteristics with the other coronaviruses and also MERS and SARS. And so one of our colleagues, actually, who works in that program, received a call in late January, early February from a physician in Washington State. And this is a case that's already been reported. It's been published in a journal, but a patient returned from China, had been visiting family there, felt ill on the way back, recognized that it was an epidemic ongoing in China and was a possibility that he might have the new coronavirus. And so he contacted his physician, who astutely contacted the public health department and the CDC, and he was diagnosed fairly rapidly with SARS-CoV-2 and observed for in this disease, and instead of recovering, he actually progressively got that he needed an emergency intervention. So called our compassionate use program and spoke with my colleague. I think she was after dinner one evening, and because the compassionate use program, because we had a supply of remdesivir, and because the compassionate use program was already in place, she could contact the FDA and have authorization to provide the medication within hours. So she got a call at 9 in the evening. We shipped the medication at 1 in the morning. And the patient received it the next day and responded favorably. Whether it's because of the medication or not, who knows? But nonetheless, it was a good outcome. And that began basically for us, was essentially a larger compassionate use program that we expanded across the globe. In terms of providing medications to patients, compassionate use is not the most efficient way to do it. Every country has their own regulations. They vary, they're quite complex, they're time-consuming for both the physicians who want to provide the medication as well as for regulatory authorities and the company that provides them. So, but nonetheless, that program proceeded until we'd actually treated several thousand patients by compassionate use, and it consumed a good portion of the time, man hours at the company. People were pulled off of all sorts of other projects to try to meet these very critical needs of patients who were requiring an emergency intervention. There's another form of compassionate use called expanded access. After we had treated a certain number of patients under compassionate use, we had the opportunity with the FDA to discuss providing expanded access. This is a program that, again, Tom alluded to before. Larry Kramer and Tony Fauci and others helped to establish this type of program for HIV back in the 1980s, early 90s. And this is essentially was the foundation for this type of program. And this allows you, instead of approving patient by patient, it allows you to approve an investigator at a center for providing the drug to patients, and then the investigator can select those patients that should receive it. It's still done under a study with informed consent, et cetera, but it's a much more efficient way to deliver the medication. And then that went on for some period of time and then was followed quickly thereafter by an emergency use authorization on May 1st. Which again, as Alan alluded to earlier, allows the FDA to provide a medication under emergency circumstances that's potentially could provide benefit, but hasn't been proven to provide benefit. And in the US, remdesivir still has not been proven to be safe and effective. It's not approved by the FDA. It's only available under emergency use authorization. It is actually, however, approved in Japan and was a few weeks ago. So that was our first official approval. And we have various sorts of regulatory approvals in different stages around the globe. But we had an initial stockpile of about 5,000 vials to address the Ebola epidemic. However, as soon as we recognized that, that we were going to need more, we did two things. We had some what they call API, active pharmaceutical ingredient, which is the product, but not in its final form. Remdesivir is given intravenously. It has to be sterile, administered and prepared in sterile circumstances. So we began immediately converting the existing active pharmaceutical ingredient to final product, and then began the process of beginning de novo manufacturing at our own facilities and quickly establishing a partnership with other companies, including our generic partners across the globe. And that process is still ongoing. So unfortunately, these medications are quite complex. I think remdesivir is, for those of you who are chemical engineers, I think it has 5 chiral centers. I may be mistaken about that, but that basically means it's a very complex molecule to manufacture reliably. And so normally it would take close to a year. It's a sequential process involving 25 steps. We've been able to shorten that to about 6 months. And so we have more supplies coming on hand from our own production facilities and from our collaborations over the next few months. And there is an open letter on our website that our CEO provided describing in detail this process and a commitment for us to manufacture over 1 million doses by the end of the year. And we've also made a commitment to donate the first 1.5 million doses to both clinical trials and also for patient care to address the important public health emergency that currently exists. So I think I'm sorry that's kind of long-winded, but that's sort of the story. So hopefully that covers it. If we had to start from scratch, the normal process for developing a drug, we discovered remdesivir in 2010 or 11. And so the normal process for developing a drug takes about 10 years on average.
Thanks, Jim. Really, really amazing, and certainly the donation of the treatment. It just, I think, kind of speaks to the global effort that industry and others are coming together. You know, when I heard the anecdotal story about the kind of the first patient who received this under compassionate use, it kind of brings me back a little bit to the digital side and thinking of sort of the work that James has done in terms of developing this tracing platform. And I was trying to think of analogs we can draw because we have this clinical and technical kind of voice on the panel today. And when I think of clinical trials, we sort of have the number needed to treat, right? So the number needed to treat to show benefit. Which is, you know, considered an epidemiological measure. And so I want to kind of, James, in hopes of kind of bringing us together around kind of a central metric, if you will. If someone's going to download the app, what's kind of the number needed to trace, right? So if I need to treat, 5 patients to get, one that shows the benefit of treatment, how many do I need to trace? And I know you gave me some numbers on our prep call, but I think maybe for the listeners to really understand, if myself and my 5 friends download this app and we all have it, how is that going to protect, the city of Santa Monica? What needs to be sort of the penetration of this technology to be effective?
So it's probably easier to answer that if I give you a little more context and elaborate a little bit on what Tom started to talk about, which is contact tracing, which is a very well-established technique for dealing with epidemic diseases. It's been applied for hundreds of years, pretty much manually. Someone become sick, you interview them, you ask them what they've done over the past few days, who they've been in contact with. You go find those people, see if they're sick. If they're not sick, you ask them to go into quarantine, into isolation for a suitable period of time so that they don't spread it. The basic idea is not that the first round of infection is terminated because it might be too late for that, but it doesn't go any further. You basically cut the chain of spread at that particular point. It's been done very successfully for a lot of diseases, but there are a couple of things that are different about COVID that make it more challenging. One of which is, of course, the scale. Thousands of people falling ill every day in a country is a big challenge, even for a sound medical system that has a lot of resources that's prepared. And only a few countries really had that, and they tended to be countries that had the experience with SARS. In fact, I don't think any of the countries in Europe were ready for this, and it doesn't look like the US was either. The other thing about COVID is that you're infectious for a couple of days before you show symptoms. It's 2 or 3 days, and you can be spreading the disease and feel perfectly fine. And there's no reason why you would I suspect that you had it, which makes it very challenging to do contact tracing because somebody could be out there in public and spreading the disease and not even know it. The third thing is that with contact tracing, there's always a very large amount of unknown. How much time were you in contact with people? Who were you actually in contact with if you were in a public space like a bus or a train? Because you don't know those individuals, you might have been on the train with them for a couple hours and they're likely to have been close to you. So the interesting aspect with digital contact tracing is that you can alleviate some of these problems. Scale is less of a problem. If you can get a significant number of people to use it, then it scales very seamlessly. The computational infrastructure is extremely lightweight. The apps are actually very simple to build and to distribute. The back-end infrastructure is trivial, and it works fairly well. There is the medical system around it, which is not easy to scale, which is actually very difficult to change so that it knows how to use digital tools. But once you've done that, you can do this as an adjunct to conventional contact tracing. The other thing that it works very well is that you don't have to get 100% of the population using it. It's like many of the mitigating factors, things like face masks, social distancing, washing your hands, each one of which contributes to reducing the R value, the spread of the virus. So none of them are sufficient by themselves, but if you put them together, you can push the R value below 1, and so the disease doesn't grow exponentially. So contact tracing is part of this, so that it doesn't stop the first generation. So you and your friends were together. One of you was infected. You were all together for a sufficiently long period of time at too close of a distance. And unfortunately, it's likely that several of you, probably not all of you, will become infected. And so how do you notify these people that they were infected before they go out and infect other people? And the answer is, with the app, it's actually very simple. That the app records who you were in proximity to. Proximity is a definition that the public health authorities in a particular country decide upon. In Switzerland, it's within 2 meters for 15 minutes or more. Other countries have chosen different definitions. In France and Italy, I think it's 1 meter for 15 minutes or more. And all this information is kept on your phone. It's not shared with anybody because it's not relevant. If you, for instance, were diagnosed with COVID, you were given a COVID test and you were in Switzerland, you would be asked if you were using the app, and then if the answer was yes, you would be asked if you would voluntarily upload the information that your phone had been broadcasting over the period of time that you were potentially contagious. And that information goes to a server and is shared with everybody else's phone. And I, on my phone, would download your information, and I would match it against all of the Bluetooth beacons, the identifiers that I had seen over the past couple days. And I would look at that information and I would say, was I within 2 meters for 15 minutes or not with Jennifer? And if the answer was no, then there's no real issue there, and potentially there's no infection. On the other hand, if we were within that distance, you would get a pop-up on the app on your phone that in Switzerland at least would say, call this phone number. And that phone number would take you into the public health service in Switzerland, where somebody would talk to you, they would interview you, they would basically give you information about this and advice that you should go into quarantine for a period of time so that you don't spread the disease further along. So it's a combination of digital, which is the sort of mechanism, and the public health system, because the people have to be there on both ends. Somebody has to give you the COVID test in the first place and ask you to upload the information. And then if you're told that you might be infected, you have to be able to talk to somebody who can give you the information you need about what to do about it, how likely you are to actually be infected. In Switzerland, it actually would then connect you to a whole set of benefits where, for instance, you would get a medical certificate which would ensure that you didn't lose your job, that you could collect 80% of your salary while you were in quarantine. So the whole system is actually very complicated because it's not just a digital app. It actually has to fit into the context of the existing health system and work with a health system that really wasn't ready for this even a month ago, let alone right now. And it's been a real challenge to bridge the simple, clean digital world and the much more complicated real world.
Yeah, thanks, James. I have a follow-up question about the kind of the third leg of that partnership and how this would partner with big pharma. For those of you that are listening, I want to remind you that Slido is open and using the code provided at the beginning of the panel, and we'll be transitioning to interactive Q&A at the hour mark. So I do see questions coming in. Thank you for those submitting those questions. And actually, I think our next question ties into some of the early remarks that I'm seeing. James, you talked about sort of society almost engaging in a partnership, right, with each other and with our health systems to do our part to potentially use proximity tracing to help sort of scale the ability to identify early chances of transmission. And Alan, in particular, I know that Roche is a partner of the NIH Active, which is the National Institutes of Health Accelerating COVID-19 Therapeutic Intervention and Vaccines Partnership, which is quite a mouthful. So I think Active is a good acronym to go with. And what role do you see public and private partners playing in coordinating the response to COVID-19? Because certainly I know that I think there are over 19 industry partners that are working now within the framework of our government to identify promising lines of investigation. Alan, do we have you?
Muted.
He's muted.
Oh.
There you go.
There you go.
So I got it. I unmuted, somebody helped me, and now I'm unmuting again. So Active is an exciting partnership for therapeutics, but I'm the diagnostics guy. So let me talk to you about public-private partnerships with diagnostics, because just like Tom said in the HIV epidemic, it changed the way we thought about drug development. I think this is the time to think about diagnostic development and topics such as biospecimens, real-world evidence, quality. We need a frank discussion about testing quality. That's been in the news. I think We have quietly and sort of complacently assumed that testing quality was to be assumed, but in fact, it's a real thing, and there are discussions taking place regarding that. But talking about how we work with the FDA, the CDC to bring tests to market, how we use real-world evidence, sort of a common topic that this pandemic has brought up is informed consent. How can we do informed consent better? How can we accept alternative forms of evidence? You know, things derived from cell phones, the Internet of Things. Can we have completely remote, low-contact clinical trials? And how does one deploy that? How do we get blood specimens remotely, sort of self-administered blood specimens? All of that is on the table. And finally, we're having broad and serious discussions about all of this and how it affects how diagnostics are developed. Jennifer?
Thank you, Ellen. I think that, Tom, is a nice segue, as you touched on a little bit earlier, but I'd like to expand on in terms of, Jim spoke briefly about the emergency use authorization, the EUA from the FDA being an enabling regulatory policy. What other policies, regulatory, global, or otherwise, have enabled scientific validation and speed to market or collaboration, much as Alan just talked about for COVID-19
well,
research?
I think one thing that a lot of Americans aren't really aware of and don't fully appreciate is the incredible investment, long-term investment that the United States government has made in HIV. And it's this kind of long-term investment that then provides an infrastructure for other diseases like COVID that come along. So the United States government did two things that I thought were very clever. One is they provided this is since 1986 now, they provided academic medical centers around the country grants to bring foreign physicians to the United States to earn advanced degrees in public health. At UCLA, we focused on Asia. We focused on China, Vietnam, Southeast Asia. Other institutions focused on other countries. This cadre of people trained under this mechanism, they went back to their home countries. They now provide the in-country intelligence that allow us to attack COVID on a global scale. That's a long-term investment. The other long-term investment were in the AIDS clinical trials groups. So again, the NIH has funded a treatment network, a prevention network, a vaccine network, and a microbicides network. And these are international clinical trials groups. And this is a long-term effort because you have to have the trained people on the ground, you have to have the quality control, you have to have the laboratory capacity. And this capacity is now being deployed for COVID. Mm-hmm. It will be deployed for treatment studies, it will be deployed for prevention studies, it will be deployed for vaccine studies. It's ready to go. You can't create that in a week. It's not just a simple policy, it's a long-term investment. And so I think we all recognize that COVID won't be the last animal to human viral transmission. We have a whole history of them. There are going to be more as humans encroach on animals and in various ways. I think that the thing I would emphasize is a long-term investment in infrastructure that is ready to go when these emergencies emerge.
Thanks, Tom. And I think, speaking of enabling technologies, James, going back to you in terms of contact tracing, a theme that I've heard in this discussion is the ability to execute trials quickly. And many on the listening end of this have probably heard about the human challenge trial, which will come up because I see a lot of questions about vaccines in the queue. But James, how do you think about perhaps marrying your platform with being able to kind of geomap, right? Where cases are happening, and how might we be able to kind of combine your technology with clinical trial, as Alan spoke about, remote clinical trial deployment, management, some of that real world evidence that we've been hearing about.
So with the apps that we built, and actually the apps that are built on the Google and Apple platform, you can't geomap. You cannot collect location information and use the Google and Apple interface. That was a decision made to preserve personal privacy. Location information is extremely intrusive about people's history, where they've been, who they've been associated with. And it's also not necessary for the epidemiologist point of view for doing contact tracing. It's fascinating for epidemiological research, but it's not necessary for contact tracing. And the app we designed was designed for exactly one purpose. It was designed for notifying people that they'd been in contact with somebody who was diagnosed with COVID or some other disease in the future. And because of that, it's extremely privacy preserving. There's no leakage of information. You have no idea who you were infected by. No one who you were in contact with can be determined from the information that's collected on your phone. So it can be deployed with a great deal of confidence by people. It's also got another very nice property, which is at the end of its useful lifetime, when the vaccine comes out, it's what we call self-dismantling. Basically, you stop using it, the information becomes stale, it is of no value to anybody. And so there's no privacy information that's left around that somebody could use for other purposes. Having said that, we are also recognized that the epidemiological information is extremely valuable for research and for other types of studies. And so what we envision is that that functionality is coupled in the same app or in a similar app with the notification functionality, so that people are given, with informed consent, the opportunity to participate in epidemiological research by giving more information than is really strictly necessary for contact tracing. So you could be asked, could we record where your location is, and it's only going to be used for this particular purposes. And, that's a decision. It won't affect whether you get notified of contacts or not, but it might help for epidemiological research in the future. And I think it's very important to separate the two, because if you install an app and the first thing it does is it says, can I use your GPS? Can I track your location? A lot of people are going to stop at that point and say no. And if you don't have very high participation, these apps don't tend to be as successful. Because if you think about it, 60% of the population, which is what the epidemiologists would like to see, are running the app, then the chances that the two people that come into contact is only 36%. It's the square of 60%. So it's not even that large. And, if you reduce the percentages by a significant amount, then the app just doesn't become very useful. And we've seen this in countries. The first app that was deployed was in Singapore. It got 20% utilization, even though the government pushed it quite heavily. We've seen other apps in Iceland, for instance, get up to 40%. That's fine, but it's not as useful as getting it up to 60%, 80%, which would be much, much more valuable. And part of what you have to do that is to give people the confidence that if they're going to use it, it does one thing and it doesn't infringe on their personal privacy. And so, we're looking at trying to separate these two areas of functionality and making it very clear to people that you don't need to participate in the epidemiological research to give your location information to benefit from the tracking information.
Thank you, James. I think that's kind of a nice lead into what is also just a theme I've heard earlier in the discussion. I'm starting to see pop up on the Q&A as well. But thinking about tracing, and then immunity is obviously a big piece of this discussion. And I'm not going to maybe specifically point this one to Alan or Jim. I'll give it kind of like a tip off, whichever one of you wants to take it first. But maybe Ellen, since it might be a diagnostic question, but what % of the population needs to be tested for antibodies? I know that Roche, you're looking at producing somewhere in the order of 400,000 antibody tests per week, which is really an astounding number. Again, the science isn't there yet, but how closely do these antibody levels correlate with immunity? Do we have an idea of how long immunity will last? And these might be more general questions versus exact answers. Ellen or Jim, I'll kind of toss that one ball in your direction.
Okay, so we're now actually shipping. There are two tests here. There's the PCR test, which is a nasal swab, and it detects the presence of viral RNA in the body. And then there's the antibody test, which is a blood test, which right now we have the FDA approval for. It's a qualitative assessment of exposure to SARS-CoV-2 virus. So the amount of time that the virus is actually being shed by the body is 7 to 10 days. It's around maybe longer if you're testing stool. So that's a narrow window to hit. So that is much more useful in somebody who has active virus in their body. And then as far as assessing herd immunity, that is much more the job of an antibody test. We don't know if the antibodies that are being tested actually are of immunity. That will emerge. And again, this gets back to my point where you're actually watching the evidence base. If we had our choice, we would wait 5 years and then launch our antibody test when we have all the evidence to support a claim. But in the interim, we're working on what's called neutralizing antibodies, and these tend to are measuring the neutralizing antibodies in the blood with our test. These antibodies in other viruses are associated with immunity. So that is an intermediate step where we'll be saying that the likelihood of this particular antibody that we're measuring is conferring immunity. And then there will be several waypoints as we make our journey to broad statements regarding does antibody testing measure immunity. There will be small observational studies in towns and confined populations that'll start to define herd immunity, where we've quantified the exposure, we've quantified the contagious capacity of that population, and we can watch the interaction. So it'll be an emerging process. This is a long-winded answer of yes, we've seen a variety of estimates of what to test with very good models, but also as a modeler, you always let emerging data and new data change your model. So that's my answer. I don't know, and I'm sticking to that.
Kim, do you wanna take a toss at that one as well?
No, Ellen's exactly right. We're just beginning to learn about what actually the natural antibody response is to this particular virus. And as Ellen mentioned, not all antibodies are protective, and the neutralizing ones have traditionally, with other types of viruses or vaccines, sort of been thought to be the ones that are protective. And oftentimes, when a new vaccine is evaluated, you look specifically to measure the neutralizing antibodies. But there are unfortunately also other examples of certain types of vaccines that induce other types of immune responses, some of which can actually make the infection worse. Was first described many years ago with respiratory syncytial virus, and more recently, actually, even one of the SARS viruses had the same sort of thing observed in some studies. So it will be very important, as Alan said, to try to identify what type of immune response are being generated, whether the antibodies are protective. And then I think we'll have a better idea of how many people need to have antibodies to actually say that the population might have some level of herd immunity or protection. And Tom, you want to address this? I know this is something you would comment on as well, I believe.
Well, I was going to, I think the herd immunity issue, we hear a lot about herd immunity. It's difficult to achieve, even when you get a good, strong antibody response and with a very effective vaccine. I mean, it really takes a large proportion of the population to be vaccinated. And I think sort of picking up on what you and Alan are talking about, and actually James's point earlier about the fact that you need, in order for any of these things to work or be deployed, you need the trust and cooperation of the affected populations. And I think as we go down this journey now with greater scientific discovery, some things are going to work, others are not going to work so well. And how do we communicate that to the public in such a way that it doesn't destroy trust in the process? We have a strong tradition, certainly here in LA County, but in the country, of anti-vaxxers. And so achieving herd immunity, in fact, my veterinarian, I talked to her about that, and she says, well, yeah, I have to deal with that with people's dogs too. And so we have to, the communication is going to be challenging. How do we communicate? Yes, we made progress. Yes, this is pretty good. Yes, this is advanced. No, it's not perfect. And not have people turn in the other direction. All of it requires cooperation.
Yeah, I think maybe a follow-up to you, Tom, while I have you, just kind of thinking about, distinguishing for our listeners between prevention and treatment trials, thinking as, vaccine trials as prevention trials. And, can we run a vaccine trial without a known rescue therapy? What are sort of the ethical and other scientific otherwise implications for pursuing those kinds of trials?
Well, I think there are two ethical principles there. One is you cannot test a product of any kind on a population unless that population is going to also benefit from the results, and that is have access to those results. We learned that in HIV. You can't do things in Southern Africa and say, okay, now let's apply it in the United States and forget about the Southern Africans. So that's one principle. The other principle is you just have to be able to provide standard of care. And so that anybody, say, who's in a preventive vaccine trial, and placebo would have the original initial trials would be placebo controlled. There are going to be a certain number of people getting infected, and we then give them whatever the standard of care is, and we have to apply that. But it may not be in a curative treatment at that point in time. It just has to be available to them.
Thanks, Tom. And then looking at sort of the questions coming in, maybe for you specifically, Jim, a question about remdesivir. Will remdesivir be studied or used as an effective treatment once tested positive as an early therapeutic cure, thereby mitigating effects? So maybe you can kind of talk about, I think, the specific patient cohort that this is currently indicated for under the EAU. And then specifically the question is around it being a potential early therapeutic cure or mitigating effects with early intervention.
Yeah, no, that's a very good question, and very rational, because remdesivir is an antiviral agent. And I think, as was mentioned earlier by James and others, the virus replication begins very early on, even in the asymptomatic phase of the disease. And then usually by several days after the infection, clinical disease begins, then the virus load in many or most patients will decrease. So for an antiviral agent, we found with other types of viral diseases, including respiratory diseases like influenza, this was a very classic example with oseltamivir or Tamiflu, that the sooner you treated a patient after they were diagnosed, the more rapid was the clinical response. And we could measure it in hours. We did actually do those studies, and we found that if you gave Tamiflu within 6 hours or oseltamivir within 6 hours of treatment, the recovery was very rapid. If it was 12 hours, it was less, 24 hours less. And of course, the labeled indication for the drug suggests that it shouldn't be given past 48 hours, although in some hospitalized patients, there's some rationale for doing that. But in any case, clearly, so administering it early on would make good sense. And we actually, Gilead is planning a study to look at that. So that'll be one of our next studies, is to do an outpatient trial looking at patients with earlier stage disease, because the current emergency use authorization as for patients with more advanced disease. So we hope to do that. And in more advanced patients, actually, Alan mentioned earlier that Roche now has approval for their monoclonal antibody, and I think there's at least 2 or 3 good uses for monoclonal antibodies in COVID. One, of course, would be for treatment, and at least for another emerging virus, Ebola, monoclonal antibodies turned out to be very effective for treatment. They also have, in some circumstances, potentially be used for prevention. And there are efforts ongoing now for various different groups to identify individuals who had COVID, who have an appropriate B-cell response with the right kind of antibodies, and to clone those and make monoclonal antibodies that could potentially be a therapeutic. And then, of course, the product that Roche has, which is treats the cytokine storm, or the result of the viral infection, which induces the release of many different cytokines, which is almost a different phase of the disease and requires a different type of intervention. So I think we actually have Roche has, and Alan wants to speak about it, but has initiated a study that will combine remdesivir with their own drug to try to look at exactly that in patients with more advanced disease. I don't know, Alan, if you want to comment on that at all, or?
Well, actually, I would defer that to my pharmaceutical colleagues to talk about that.
Well, our current CEO used to be yours, so he's.
You're welcome. You're welcome.
Yes. So, but yes, that's one of the next studies that's in queue, and it's due to start, I think, in June. So we're very much looking forward to that, and also the NIH. The initial studies, placebo-controlled trials of remdesivir, showing some benefit in reducing hospitalization, said that their next effort is going to be to combine remdesivir with agents that will modulate the immune response. So it's a very good question and very important issue that needs to be addressed.
Thank you to you both. I think that kind of ties in nicely to what is our most popular question currently, but it goes around exactly what I think you guys have just spoke about. So if there, if Tom or James, you want to collaborate as well, maybe on how tech is getting a big tech or tech industry is getting involved. But the question is how much collaboration or intellectual property sharing is happening between big pharma and other life science industry members? And are there any consolidations of skills or resources that would help accelerate, vaccine development or otherwise?
I think Jim or Alan perhaps would be better equipped to answer that question.
Yeah, so in the diagnostic space, I look at the public-private partnerships for the development of therapeutics. Roche has a very large, Roche Diagnostics, very large global footprint with our analyzers, and we partner with many pharmaceutical companies when a precision medicine intervention becomes necessary. And we partner with them in development. Often the antibody or the test that is used to create the cohort for the clinical study is not suitable for industrial level production and widespread deployment in the medical community. So we'll alter that diagnostic strategy so that it can accompany the companion therapeutic out into the marketplace. So we work very closely with many pharmaceutical companies to develop companion diagnostics. And the other thing we do is after the development phase is done, we'll also partner with those same pharmaceutical companies to educate the medical community about the appropriate use of the drug and the companion diagnostic. Regarding intellectual property sharing, specifically in diagnostics, we make reagents for our competitors. We buy reagents from our competitors. There is a lot of commingling of intellectual property and components. Our our devices are very much like airplanes, where there's pooled intellectual property inside of those very expensive robots. So I think that's common in the diagnostic industry, and certainly this enhances the partnering with the pharmaceutical industry.
Thank you, Alan. I'd like to switch gears just a little bit. A question coming in from George Abe, one of our professors at UCLA Anderson, who's an expert in innovation as well as life sciences. And the question is for James. It's around, I think we talked a little bit about before. I heard the number of the 60% of the population having contact tracing applications installed. The question is what percent of a population is required to produce high confidence intervals? And specifically around Switzerland, do the Swiss have sort of a public health standpoint or a target percentage that they're looking to meet?
That's a very good question. The answer is for contact tracing, there isn't a very good basis for doing confidence intervals. If you look at the traditional manual contact tracing, you would say it's extremely inefficient. You put a lot of people into quarantine who never get sick. There was a recent paper in the Lancet about one of the first cases that occurred in Germany where they did traditional contact tracing. The person traveled to Wuhan, came back for business to Germany, met with people in the company over a few days before he fell sick. I think they eventually put about 200 people into quarantine and 7 of them fell ill. So by any other standard, that would be seen like a pretty poor return on investment. But if it's stopped, an exponential blow up at that point where it was still a very small number of people, then maybe it was a good trade-off. And so we're actually looking right now before the app is deployed in Switzerland, how we're going to evaluate it. So we've been working with statisticians, biostatisticians, to try to figure out what a robust way of evaluating the deployment of the app and trying to come up with an answer to the obvious question, was it successful or was it a good idea? And I can't tell you that we have a very clear answer to that yet, but we're very much aware that it is a very relevant question, and one that there right now is no experience that would let us put hard numbers on.
Thank you, James. And kind of just as a follow-up to that question that Alan fielded earlier about sort of the collaborative efforts within industry. I know you were at Microsoft for many years. How do you see the tech industry getting involved? Certainly we've seen Google and Apple making an entrance, but what are some other ways that the tech industry might get involved in the response to COVID-19?
The tech industry has been great. In various countries, take it, for example, Germany, SAP, which is the largest tech company in Germany, is building their app for them. In Canada, Shopify, which is a large, fairly successful startup, is building their app for them. A lot of the tech companies have wanted to get very much involved and have jumped in and volunteered their people's time and their people's assistance in doing this. And certainly the Google and Apple is a good example. They have fairly large teams at both companies who have been working very intently on this for a number of months now. And it's not a product feature. It's not something that's going to convince you to buy an iPhone as opposed to an Android phone. Hopefully, if you have one of those phones, you'll use it, but it's not a sexy feature that's going to sell more products. And they did it because I think they both believed that it was the right thing to do. And actually, even more remarkably, they believed that they had to do it together, because it wouldn't work if there was a solution that only worked on iPhones and there was a solution that only worked on Android phones. You would have two separate worlds, and both of them would be smaller than they needed to be. And the collaboration actually is quite remarkable between two fairly fierce competitors. We've been sort of at the point where they come together, and we've seen it. It's a challenge from both companies' point of view, but I think both of them are very much committed to making it work.
Thanks, James. I'd like to switch gears just a little bit, and maybe a question for you, Tom, because I appreciate your mention of the events going on across the greater country currently. And question here is with the response, with the recent protests and inequalities, how do you prioritize access to vaccines in an ethical and equitable manner? And I think you spoke a little bit earlier about sort of the socioeconomic differences and prevalence of COVID-19 amongst different patient populations. So I kind of, globally, nationally, or within specific high-risk population groups, what are your thoughts around the prioritization of treatment?
Well, I think it's going to be very, very important, both for industry and for government, to develop explicit access programs for heavily affected communities. We've done this in HIV. Again, I mentioned the President's Emergency Plan for AIDS Relief, the Global Fund for HIV, TB, and Malaria. These are hugely successful endeavors. They build on the philosophy of health diplomacy, and that is the best way that we can make for a better world and make for a peaceful world is by helping one another in terms of health. Health is vital. It improves the economy, and the economies improve health. So I think there have to be deliberate strategic programs addressed to the affected communities. And this has to move along two lines of access, but also lines of communication, but also lines of listening to those communities. Those are the communities that are most readily susceptible to thinking, well, this was a human creation designed to get rid of us. And these stories about how there could be side effects to them. They could be very rare side effects, but there could be side effects to them, and there will be. And so engaging with those communities in a deliberate strategy to figure out how to do that is going to be essential. We've done this globally, certainly with sub-Saharan Africa and other low and middle income countries. We're trying to do it with HIV. The largest growth in HIV infections is occurring among African American men who have sex with men. And it's got to be engagement with that community that's going to provide a solution and access to those life-saving and preventive technologies that are available.
Thank you, Tom. I'd like to, I do see a question that kind of in the queue, and I think it'll sort of tie up with one of our pre-submitted questions, really about the balance between, really, I think you mentioned communication, which is timely, because how do you manage expectations of the public around vaccine development? I think what I've heard numbers, recent JAMA Journal, Journal of Medicine podcast was 2 years minimum to vaccine. But how do we balance expectations of the public around these treatments, either from the timelines or kind of the side effects or benefits, as you just mentioned?
Public Health 101, clear, consistent communication, and tell the truth. those are the bedrocks. And engage actively with affected communities. There's just no substitute for those things. I think it's, we hear 1 year we're happy about the immune response with in the eight subjects that of the one product. And this raises great expectations, and the stock market goes up, which I'm happy about. But it's and yet also our history with HIV and HIV medications is it took a while to get where we are. It took about 20 years to get to a pill a day with no side, really a great side effect profile. That's a great advancement. It took us 20 years. So I think the other day, talking to somebody, and she was saying, well, gee, the flu vaccines don't work. Well, I said, well, they do. They're not perfect, but they work for 60 to 80% of the population, and that's a good thing. So how do we communicate those things? But Public Health 101, clear, consistent, truthful communication. It's got to be on the front burner.
Thanks, Tom. I'd like to close out with a final question, really kind of, I think what we sort of forecast a little bit in the webinar description around sort of the new normal for each of your respective industries or roles in addressing this pandemic. Really, I think it's pretty astounding that the folks on this call have really responded with viable products in literally months from a kind of concept or existing product to market, which is outstanding. And so, how will the healthcare industry fundamentally change? We saw an announcement from the FDA today to kind of expand, kind of continue to prioritize COVID-19 treatments. So if each of you could just kind of take a minute, maybe starting with Jim, and working our way down the line on how the new normal in response to COVID will affect our lives.
Working from home, that's been my story for the last 2 months or so. It's actually worked better than I figured it would, but I think there's really no replacement, in many circumstances for face-to-face interactions, and particularly if you don't know someone and to get to know them, I think really interacting with them one-on-one is very important. So I'll look forward to hopefully getting back to that at some point in time. But we have a lot to go through between now and then. Fortunately, 10 years ago, I don't know if this technology would have been available and we wouldn't have been able to continue to work together as we have. You think back, I think the iPhone was 2007 or something like that. It's just incredible, the technology that's been developed in this period of time. But it's allowed us to at least to continue some significant level of progress, and hopefully that will continue and we'll build from there. But we're all working together on it. Hopefully we'll get there.
Thanks, Jim. James?
Well, my day job is education, and obviously that's been a little bit disrupted by COVID. I don't think the students found working at home to be much of a substitute for going to classes and being around the other students. And we hope that in the fall we can handle it. I don't think it's going to go away, so we're going to have to rely on the better education for the students and applications like the contact tracing to try to keep it under control until the vaccines come along and that it becomes less of an issue. But at the same time, what you said about working at home is also true. Along with a couple of other professors and some grad students, we essentially did a startup, which in less than 3 months was able to work with a couple of large companies and ship 1 billion copies of what we designed on phones throughout the world. So that's quite something for a bunch of academics to do in that short of time, when we literally have not even gotten together as a group over that period. So it really does say something about the technology that is available to people now. And I think that is really going to be a change, that there's no real reason to bring large groups of people together to do certain tasks anymore.
Yeah, thank you, James. Tom?
Well, all my research is in Southern Africa, and my United Club card expires in 2099. I'm a permanent United 1K. Spent a lot of years of my life flying around the world, which has been incredible. That's actually been one of the real benefits of this work, is getting to know people in other countries, other cultures, how they think and how they live, and making friends and so on and so forth. Also, we spend a boatload of money on conference calls. And the new normal for us, maybe we don't need to fly around the world so much and say some of the same things at conferences. We can make our work more efficient and more productive and more collaborative. I think one of the lessons we've learned from HIV is that strong global networks are important. And I think the technology makes that all the more feasible and reachable. And so hopefully the new normal, well, maybe we won't affect the climate so much and still get good work done.
Thank you, Tom. Alan?
Oh, I think this may be a significant event for the diagnostics industry. The whole concept of rapid diagnostic development and how we can change the course of illnesses in populations is finally getting a really good public hearing and a good hearing in government. For Roche, we're just getting started. We think in terms of the diagnostic journey of people, and we still have point of care solutions to develop. We have solid phase solutions to develop. When vaccine trials come out, they will need diagnostics. When therapeutics come out, there will be companion diagnostics. So it is an exciting time to be in the diagnostics industry, and we're ready to go. We're just getting started.
Thank you, Alan. I'm going to ping you in a year to do a case study on all the great work that you're doing in the diagnostic space, perhaps. I think what Tom said about communication is so important, and kind of the power of communication. And I'd like to thank all of you for joining us at Anderson virtually this evening, because certainly the unique skill sets and expertise that you've all brought to this call is really kind of a unique experience for our alumni and Greater Anderson community to hear kind of the really frontline advances in products that are being launched to address the effects of this pandemic. So I thank you at this uncertain time, culturally, as well as clinically, biologically speaking, in response to COVID-19, for coming together and for sharing with our Anderson community. So thank you, Alan, Jim, James, and Tom, and look forward to reading more about the great work that you do.